Hello, everyone, and welcome. I'm Sarah Van Driest. I'm the Director of Pediatrics for the All of Us Research Program. And I'm welcoming you to our scientific session 2A, How Our DNA Shapes Health Across All Communities. We can go to the next slide, please. Before we get started, I want to share some housekeeping. Closed captions are available throughout the day. To turn on the closed captions, go to the CC icon in the top right of your screen and choose your preferred language. If you run into any technical issues or need an accommodation, please email allofuscienceday@pyxispartners.co. Next slide. As we get started, we want to create an open, respectful, and engaging space for everyone. To ask a question, select the Q&A option on the top right of your screen. Type your question and submit it. The Q&A is private and monitored by all of us staff. Questions submitted through the chat box will not be monitored. The chat is public, so please be mindful of any personal information that you share. As I mentioned, Research Panel 1 will focus on how our DNA shapes health across all communities. And I am delighted to introduce our panelists. Please go to the next slide. Please join me in welcoming our three panelists, the first of which will be Abhiram Dinesh Kumar Binoma, who is a graduate student at Queensland University of Technology. Our second presenter will be Alexandra Miller, who is a MD PhD student at University of Arizona. And our third speaker will be Adriana Bisbal, an assistant professor of biology at the University of Houston downtown. Again, during the presentations, if you have any questions, you may put them in the Q&A section of the chat, and we will get to them during the Q&A portion at the end. And so let's begin today with Abhi.
Can we navigate to the slides, please? Yep. Good morning, everybody. Myself, Abhiram. I'm doing my PhD in Queensland University of Technology, and today I'll be talking about my PhD research topic, which is the hidden genetic links between heart, kidney, and diabetic health. Next, please. Next. So let's start with a story, which is not a random story and it's not a build-up story as well. It's a story of my family and it's a story of my father. Next, where he's of age of 32 and he's not having any of the preconditions and he is going to hospital for a regular checkup, and later it has been diagnosing that he's having a high glucose level in his blood, and basically it's a high sugar level, and doctor has been diagnosing that with also type 2 diabetes, and from there onwards he started having the type 2 diabetes medication. Next, so after having the type 2 diabetes medication for 2 to 3 years, he started having an excruciating pain on his left leg, and we are taking him to the hospital for this. And the doctor says that he is having a stroke on his left leg. Basically, it says that there is a blood clot on his left leg, which is not allowing the blood to properly flow to this left leg. So basically, the doctor is suggesting that the only way that we can do it is do the amputation, the leg amputation. And considering his age as well as the young family, the doctor is saying, okay, how about like going with the medication? If in case the medication is not working, we have to amputate his leg. So we have decided that let's go with the medication, and thank god, and it like well, finally the medication, the situation got improved, and we don't need to do the amputation. So basically he started having the type 2 diabetes medication, and now he started having the cardiovascular related medication as well. Next, so after having all these medication for like 2 to 3 years, and now it's been 5 years, and he is not able to sleep properly. He's while he's sleeping, he's having a discomfort on his back, and we're going to hospital again, and we have been find that we have been diagnosed with a starting of renal failure, basically chronic kidney disease. So what you have seen here, next, so what you have seen here is the interconnection between these three conditions, basically starting from diabetes, going to the cardiovascular disease, then to the chronic kidney condition. So this is a story, and it's not a story of one individual or a one family. It's a more than 600 million individuals family will be able to say the same story, because these three conditions are co-occurring. Next, please. So the story doesn't end there. So my father is going to pass on his trait and behavioral everything through his DNA. So also along with that, he is going to pass on the susceptible of getting these conditions as well to me. So he is going to pass on to me, and I'm going to pass on to my children as well. So we need to break this chain in somewhere. Next, so what we are trying to show here is, or to identify from this study is, to identify what, like, is there any, like, whether genetic is playing any role in this, in the co-occurrence of these conditions. So either like the multimorbidity, is it associated with the genetics? So that's what we want to see. Also, along with addressing that, we would like to address one of the major limitation in the genetic study is the studies are more concentrated on the European ancestry results, or like European ancestry group. We would like to expand that to the other group ancestry groups as well. Next, so here what I'm showing here is the major, like the overall picture about what we have done is to, we have selected different study groups, basically different ancestry starting from European, African, as well as East Asian. So we have collected the majority of the data comes from all of us. So not only all of us, we have other biobanks such as UK Biobank, Million Meteorum Program, FinCEN, China Cadore Biobank, Japan Biobank, and Taiwan Precision Medicine. So we approached these all different biobanks, and we collected the GWAS summary statistics from all these biobanks from irrespective of each of the ancestry. So what the GWAS summary statistics tells is that how much, or the what are the genetic variants which are contributing to the conditions. Next, so what from we approach the different biobanks, and these are the major conditions. So for example, here what you're seeing is the type 2 diabetes, hypertension, chronic kidney disease, ischemic heart disease, and heart failure. These are the five conditions that we have targeted on, and we have collected the summary statistics from each of the biobanks for these respective phenotypes, and we need to collate these together. So basically we did the meta-analysis to collate the results together and come up with one results. So basically we did the meta-analysis. So now we have collated results for each of these phenotypes. Yes, now we have each of the condition. Now we need to come up with a multi-morbidity. So to address our objective, we need to get the multi-morbidity. So how we achieve the multi-morbidity is we try to group this phenotypes together in such a way that hypertension, ischemic heart disease, and heart failure. So in the right side of the figure, what you're seeing is the grouping structure, which is hypertension, ischemic heart disease, and heart failure are grouped together for the cardiovascular, and along with cardiovascular, type 2 diabetes and CKD have been grouped together for the multimorbidity. So one thing that I would like to highlight here is that we are able to see this pattern in not only one ancestry, across all the three ancestries. So this we are able to achieve with one of the specific method, which is genomic structural location modeling, and which we helped us to achieve the, to identify the genetic variants or the mutation which are associated with the multimorbidity. Next slide, please. So here what we are seeing is the genetic correlation, which tells that what is the genetic overlap between each of the conditions. For example, I have highlighted one value which is 0.65, which shows the genetic correlation between heart failure and hypertension in the European ancestry. Next, please. Along with European, we do have other ancestries as well, East Asian as well as African. So that genetic correlations is also there. And so for example here, this is one of the like key findings that I just put here, which is showing that what are the so we did the multi-morbidity GWAS, basically what are the genetic variants which also with the multi-morbidity for each of the ancestry. We could identify like thousands of mutations, thousands of genetic variants which are contributing to the multi-morbidity. So for to talk to one of the key finding here on the right side, I'm talking about the FTO gene, which is fat mass and obesity associated gene, which is known to be associated with the type 2 diabetes or related to metabolic diseases. And now from our study, what we have identified is there are 24 mutations, 24 SNPs, which are found to be associated with the with the multimorbidity in not only one ancestry, across the three ancestries. So we are able to adapt to the definition that it's not only related to a metabolic or metabolic syndrome, the FTO gene, it's also related or to be associated with the co-occurrence of these conditions. FTO is one among the gene. We do have around 16 genes in the list, which shows that are to be associated with the multimorbidity. Next, please. To summarize the results or the whatever I'm speaking, like which is the we did a multivariate GWAS to identify the genetic variants which are to be associated with the multimorbidity. So the key finding here is the FTO gene, TCF7L2 gene, which are known to be associated with the different phenotypes. Basically, like we are known to be associated with each different conditions, and now we are able to adapt to the definition or the redefine in such a way that it's not only associated with any of the each condition, it's also associated with the co-occurrence of these all the three conditions. And how this going to enable or how is going to improve in the near future's research is that it is going to improve the prediction accuracy for sure, and also it is going to improve the drug target discovery. Basically, it is going to improve in such a way that we may in the near future, instead of having each drug for each of the condition, we may in the near future redefine in such a way that one drug for all three conditions. So that's our goal in the future. Next. And before finalizing my presentation, I would like to thank all the participants who have been generously contributing their data. Without them, it is not possible at all. Next. And I would like to thank everybody who have been contributing to the research and all the biobanks, including all of us here and all the participants across all these biobanks. And thank you all of us for giving me this opportunity. Thank you so much.
Thank you, Abhi. That was fantastic. I'll remind everyone to use the Q&A to put in your questions. We'll have time for a discussion after all three presentations. And up next, we have Alex.
Awesome. Thanks, Sarah. Hi, everyone. I'm Alex, and I'm an MD PhD student in the lab of Dr. Jason Carnes at the University of Arizona. And today I'll be talking about my work on a phenome-wide association of APOL1 risk alleles in all of us. Next slide, please. In this study, we performed a phenome-wide association study, or PheWAS, of APOL1 risk alleles in individuals genetically similar to all reference populations in all of us. To calculate the number of risk alleles for each participant, we used phased haplotyping that included the APOL1, G1, G2, and N264K variants that I will define and describe in more detail on the next slide. We found that APOL1 risk alleles were carried by individuals genetically similar to all reference populations, meaning that people in our cohort from every ancestry had risk alleles. And we confirmed their association with an increased risk of kidney disease. We also found a new protective association of the APOL1 risk alleles for HIV infection. These results are important for clinicians to be aware of, as we found APOL1 risk alleles exist in populations broader than what they may consider for APOL1 kidney disease. The new protective association with HIV infection provides the first epidemiological evidence of this association and a new avenue to investigate APOL1's involvement in innate immune processes and infectious diseases. Next slide. APOL1 stands for apolipoprotein L1. The APOL1 locus is located within the APOL gene cluster on chromosome 22. The top of this slide shows a representative schematic of the APOL1 genetic locus. The APOL1 risk alleles, G1 and G2, were first identified in 2010. The G1 allele consists of two non-synonymous variants that are in near-perfect linkage disequilibrium. I will refer to the two G1 variants as G1A and G1B throughout this presentation. The G2 allele is a 6 base pair deletion, deleting 2 amino acids from the APOL1 protein. Individuals with 2 copies of an APOL1 risk allele have now well over a decade of evidence that they are at a significantly increased risk of developing kidney disease. So why haven't these variants been selected against if they're bad for kidney disease? Well, the APOL1 risk alleles are hypothesized to be another example of balanced selection, similar to the sickle cell genetic trait and protection against malaria. The APOL1 protein is known to be the serum trypanolytic factor, and the flagship paper identifying the risk alleles found that G1 and G2 help protect against infection with Trypanosoma brucei, the causal organism of African sleeping sickness. So while the ApoL1 risk alleles do increase a person's risk for developing kidney disease, they are hypothesized to have arose to protect against parasitic infection. In addition to the G1 and G2 alleles, another variant in ApoL1 called N264K was identified in 2023. When N264K is inherited on the same chromosome as G2, N264K cancels out the G2 kidney disease risk. To date, G1A, G1B, G2, and N264K have not been assessed together, and ApoL1 investigations have largely been restricted to individuals genetically similar to African ancestry reference populations. Next slide. To account for all of the variants, we used a phased haplotype approach to calculate the number of ApoL1 risk alleles per individual in all of us. Phasing assigns variants to individual chromosomes. Briefly, our phasing rules were that each G1 and G2 allele counted as one ApoL1 risk allele. If both G1A and G1B were present on the same haplotype, that was counted as one risk allele. If G2 and N264K were inherited on the same haplotype, that was then counted as 0 risk alleles. Examples of some of the haplotypes and these calculations are shown on the table on this slide. Next slide, please. For PheWAS analysis, we used standard adjustment variables and tested the association of number of ApoL1 risk alleles expressed as a continuous variable across 3,425 electronic health record-derived Phe codes. We ran PheWAS in multiple cohorts, including across the 7 genetic ancestry reference populations available in all of us. The results that I show today will be from our overall cohort that included 231,423 all of us participants who had available genotypic data, electronic health record data greater than 1 year in length, and the PheWAS adjustment variables listed on this slide. Next slide. When assessing the distribution of number of risk alleles across genetic ancestry reference populations, we found ApoL1 risk alleles were carried by individuals genetically similar to all reference populations available in all of us, meaning that people in our cohort from every ancestry carried risk alleles. The majority of individuals with 2 ApoL1 risk alleles, indicated by the red dots on this principal component scatter plot, were from individuals genetically similar to the African, European, and America's ancestry reference populations. Next slide. Our PheWAS analysis in the overall cohort yielded 26 Bonferroni level significant phenotypic associations. This slide shows a PheWAS Manhattan plot, with the x-axis being categories of the phenotypes and the y-axis being the p-value. The significant phenotypic associations are labeled on this figure. I'd like to draw your attention to the 2 most significant phenotypic associations outlined in the orange boxes for end-stage renal disease and kidney transplant. These results provide a strong proof of concept in our PheWAS approach and confirm our expectation that individuals with ApoL1 risk alleles have an increased risk of kidney disease. Next slide. This slide shows exactly the same data as the previous, but it is presented in a volcano plot, with the x-axis being the odds ratio of the phenotype and the y-axis again being the p-value. Dots towards the right, with an odds ratio greater than 1, are phenotypes associated with an increased risk in individuals with ApoL1 risk alleles. Dots towards the left, with an odds ratio less than 1, are phenotypes associated with a decreased risk or protective effect in individuals with ApoL1 risk alleles. While the majority of significant phenotypic associations were related to kidney pathology, I'd like to draw your attention to our novel result outlined in the green boxes, with ApoL1 risk alleles having a significant protective effect for HIV infection. Next slide. In conclusion, this work found that ApoL1 risk alleles are carried by individuals genetically similar to all reference populations available in all of us. This is important for clinicians to be aware of and to potentially consider APOL1 kidney disease in broader populations. PheWAS in all of us of the APOL1 risk alleles confirmed their association with an increased risk of kidney disease and provided the first epidemiological evidence of a protective effect for HIV infection. This new phenotypic association may have implications for APOL1 targeted drug development, including the potential for adverse reactions. Next slide. This work would of course not be possible without the All of Us participants, so I'd really like to thank them specifically and all of the All of Us researchers. Next slide. I also want to acknowledge and give a big thank you to all of the members of the Carnes Lab involved in this project, our project collaborators, and of course, our funding sources. A little plug is that Dr. Carnes and the University of Arizona runs a biomedical informatics consult service for anyone interested in using the research workbench, so please reach out if you have any interest in this resource. I'd also like to give a special shout out to my colleagues, friends, and family who were able to join today. And next slide. Here are my references. Next slide. Thank you so much for the opportunity to present and for your attention. I'm excited to take your questions.
Thank you so much, Alex. That is really interesting. And again, we'll be taking questions after this talk for all three speakers. So put those in the Q&A box if you have questions for them. Next up, we have Adriana. Thank you for joining us, Adriana. The stage is yours.
Thank you, everybody, for this opportunity. My name is Adriana Bisbal, and today I am an assistant professor at the University of Houston downtown. And today I'm going to be talking to you about a project I've been doing with my undergraduate students that I'm excited to share about closing the genomic diversity gap and using computational tools to reclassify BRCA1 and 2 variants of uncertain significance with a focus in the America's ancestry population. Next. So inherited changes in BRCA1 and BRCA2, also known as BRCA1 and BRCA2, increase breast cancer risk. So these two genes, the BRCA gene 1 and the BRCA gene 2, produce proteins that help repair damaged DNA. So I want you to imagine that genes are like recipes, right? That we have in our DNA. If our DNA is our sort of cookbook, these two recipes essentially encode the instructions for the final product that are these two proteins that help with DNA damage repair and have a lot of other biological functions. So when they are not working properly, they can increase the risk for cancer acquisition, especially in breast and ovarian cancer. Next slide. So in an individual that has a normal BRCA status, that doesn't carry mutations, right, or damages in this recipes for BRCA, the risk of developing breast cancer by age 70 is about 12%. Now, if you have a mutation in BRCA2 or BRCA1, you see how this risk dramatically increases. And the risk can go up to 45% for certain mutations in BRCA2 and from 55 to 65% for chances of developing breast cancer by age 70. Next slide. So when individuals, but not all the mutations are the same, right? Not all changes in a recipe can result in the same damage. So when individuals get tested for a herited or inherited change in BRCA1 or 2, you can get several outcomes for that test result. So I want you to focus on the little red alarm on the left-hand side, which is a pathogenic or a harmful genetic test result. This would indicate that the variant or the change that you have in your DNA sequence or your recipe, right, is that it causes increased risk of developing cancer. Imagine this is a situation. Can you go back, please? What's happening with the slides? Sorry. One more, please. Advance one slide. I don't know what's going on. I'm gonna take control of the slides. Here, sorry. Imagine you have a situation. I don't know who's advancing the slides. Can you please advance the slides? Thank you. Imagine you have a situation where you have a recipe swap between salt and sugar, and that would dramatically alter your risk of cancer. So therefore, you would have a pathogenic result. On the other end, on the right of the slide, you would have a benign or harmless, right? This would be a DNA change, right? Or a variant that even though there has a change in your DNA or a change in your variant, it has not, it does not cause any change in the protein function. This would be a, for example, a situation if you imagine stir to the right or stir to the left in your recipe, right? That would be a benign. So this genetic test result of benign gives you ease. You go back to standard screening recommended. A pathogenic result has clear clinical guidance, right? For increased screening and, for example, surgical prevention techniques. In the middle, we see something known as a VUS, a variant of uncertain significance, right? This means we do not know. You have a genetic change, you have a change in the DNA sequence, but we do not know what has been happening to the change in the DNA, and we do not know if it's associated with increased risk. This leads to anxiety. We do not know if it's clinically actionable. There is no clear guidance. So the question is, how do we determine which category a variant belongs to? Next slide, please. So in order to understand if a variant is associated with pathogenicity or benign, right, if it's associated with increased cancer risk or not, there are a variety of different types of studies. We can use genome-wide association studies, like we heard from our first presenter, where we look at a wide variety of diverse genomes, and we see, are they associated with an increase? Are those variants, are those changes associated with an increased risk of cancer? We can also do functional studies in the laboratory, or we can see, because these are inherited changes, if this change runs in families that are at an increase, right, where this increased risk of cancer is happening. Next slide. So one of the problems that we have is that for a lot of databases that exist, genomes of European descent dominate those databases. So if you look at the graph on the left and you compare individuals that have participated in genome-wide association studies in millions and the global population on the right-hand side in billions, we see that even though we have a drastic increase in genome-wide association studies, the red, right, European ancestry, as indicated in red, really dominates the majority of these studies. This is also illustrated by the GWAS diversity monitor graph on the right. So if we are looking for variants, right, if we are looking for changes in the recipe and we are trying to compare, is this a change in the recipe that is naturally occurring? Is this just the way that your great-great-great-grandmother used to make this recipe? Or is there a problem, right? Have we swapped the salt for the sugar? We have no way of knowing because we're only comparing, right, with just a certain ancestry. So that creates an increase in VUS returns or variants of unknown significance for individuals that are not of European ancestry. Next slide. So our answer, right, to solve this problem is using the All of Us workbench. We know that it is a very diverse database, and what we have been using is the latest release of the database that contains approximately 415,000 genomes, and we see that it includes approximately 84,000 genomes of individuals of African continental ancestry and the AMR continental ancestry. This AMR continental ancestry includes individuals that have markers in their ancestry from Native American, so from the American continent, indigenous populations, and also it is enriched for individuals that identify as Latin American ancestry or Latin American origin. And this has been the focus of our population study. Next slide, please. So what we did is that we analyzed the genetic variants, so all of the changes in those in the DNA for the BRCA1 and BRCA2 genes for all of the people in the All of Us research program. And we use computer prediction programs to identify which of those variants that were classified as uncertain after we run them through computer prediction programs were actually found to be disease causing. What we found was that if we focus on that America's ancestry population and those variants that were of uncertain significance, we found that our computational tools actually predicted that 50% of our BRCA1 and 41% of the BRCA2 VUS were likely to be disease causing. Why this matters is because each time that we reclassify a VUS, we are helping future patients by eliminating genetic uncertainty. So our work is getting to hopefully directly address health equity, but ensuring that precision medicine is serving all populations and not just those of European ancestry. So let me give you a little bit more detail into our work. Next slide. Okay, so we started with the All of Us variants, right? That contains a large cohort, right? Approximately 45 million variants, which is a very powerful database. We use, HALE genomic analysis. It's a Python based code, right? To use to pull those variants in the web-based Jupyter notebooks, and then we pulled those variants and found 4,294 in BRCA1, 7,981 in BRCA2. Then we sorted them, we filtered them for the ones that were present in that admixed American ancestry, the AMR ancestry, and that left us with 1,180 for BRCA1, 2,323 for BRCA2. Then we correlated with the ClinVar classification, right? This is a classification done by sort of a panel of geneticists that gets together when these variants are reported and says, okay, what are they benign? Are they pathogenic? Or are they VUS? So we first focus on the missense. I'm sorry. So these are changes that are going to cause amino acid change, right? So a change in the DNA that causes an amino acid change. And then from there, we focus on the variants of uncertain significance, giving us 336 VUS for BRCA1 and 776 VUS for BRCA2. The computational tools that we focus on are PolyPhen2, which uses protein structure and evolutionary conservation, and mutation tester, which is complements PolyPhen2. Next slide. And then our key findings is when we run them through, we found that approximately 50% of the ones for BRCA1 were disease-causing and benign, and 40% of the BRCA2 were disease-causing and approximately 60% benign. So in total, we have found that nearly half of those that had uncertain significance have now computational evidence that they may be disease-causing. Next slide. Here's one example, right, of one that is actually American ancestry specific. So this variant would have never been found. It is exclusive to individuals of AMR ancestry. So if we would have used databases that did not have individuals of AMR ancestry, would have not been found this variant at all. Next slide. And here's the ClinVar information showing that this has conflicting classification of pathogenicity, right? So the clinical experts, because there is not a lot of evidence for this variant, are showing it as a VUS. Next slide, please. And our next steps are, we are now essentially at step 2, right? Finding those variants that we're going to prioritize. We then hope to do some experimental validation in the laboratory, establishing partnerships with clinical labs to validate this in patient samples, and then hopefully get to the goal of clinical reclassification, where some of these variants can now get the label from VUS to either pathogenic or benign, and eliminate some uncertainty for our patients. Next step. Next slide. And I just wanted to mention that we also, part of our work is to communicate. We don't want to leave the science just for scientists. So we have a series of workshops called Cafecito para la Salud, where there are bilingual workshops where we explain BRCA inherited mutations to the community, and we talk about and demystify genetic testing. Next slide. Want to make sure that the All of Us research program wouldn't be possible without the generosity of our participants and the dedication of our researchers. And we would like to acknowledge all of the participants, especially my undergraduate students that did a lot of this hard work.
Thank you, Adriana. That was fantastic. At this time, I'd like to bring all of our speakers back for some Q&A. We had some questions submitted, and I'd like to address some of them to each of you. First of all, let's start with Alex. We got a question about how or should people find out if they have the genes associated with these comorbidities?
Yeah.
Sarah, I think it's my question. Might come up with it too.
Yeah, Abby, you want to take that?
Okay, go ahead, Alexa. If you please go ahead.
For APOL1 specifically, the variants itself don't necessarily cause kidney disease. They're just associated with an increased risk of developing it. So the field as of right now has what's called a two-hit hypothesis, where you need some sort of environmental factor, like, for example, actually HIV infection or another viral infection that would then precipitate kidney disease developing. So as of right now, APOL1 is not suggested from like a population level standpoint to get genetic testing for, because it's not a causal gene for kidney disease.
Thank you, Alex. Let me go to Avi, because I think this is also very relevant for your conversation that you started. So tell us about your talk and what genes maybe should people be looking out for there?
Sure, so we do have a list of genes though. So I would like to say that it mostly our studies both in so we found to be associated with the multimorbidity. We still need to make sure that it is causal. So with a clinical or we have to lab test has to be performed further. And to the specific question that I asked, that the genes which is how can I find whether that gene is present in my DNA or not? So basically we can do a DNA sequencing and find that out, and it's not a hard thing. But so only thing that I would like to highlight, that is yes, I can find those genes whether it is present in your DNA or not, but to make sure that these genes are causing to the multimorbidity, we still need to do further validation. Yep. Thank you.
Thank you. So it sounds like we've got a little bit more work to do on that front. What about for you, Adriana, for BRCA1 and 2, is there anything you'd like to share with those who are listening?
Yes, and I see another question about that on the Q&A as well. So I want to clarify a little bit that these genes are genes that we all have, right? So they are present in everybody. The question is, do we have a change? And is that change one that is a change that is just present, that is not likely to affect how the protein functions, right? Or is the change going to affect function and therefore increase cancer risk? So currently there are a couple of different ways in which you qualify, right, for insurance to test you for BRCA1 and 2. And a lot of those are a little bit less clear, because one of the items is, do you have a family history of breast cancer? And that should be very clear, right? If you qualify there, then you usually qualify to get tested to see if you have a pathogenic mutation in BRCA1 or 2. But one of the other ones that gets asked in the family history is, do you have any ancestry that is associated with an increased risk? And this is sort of the purpose of why we're doing the studies and why all of us exist, because we know that, for example, individuals with an Ashkenazi Jewish ancestry have an increased risk. There are other populations that are at an increased risk of carrying mutations that are associated with increased risk in cancer in BRCA1 and 2. But because we haven't studied so many individuals with African ancestry or AMR ancestry, the answer is we don't know. So part of our work is also to figure out what the prevalence is. And I do know that there are a lot of clinicians that are pushing for maybe this testing should be more offered. I know that for Latinas, for example, it is the population that is currently tested the least for BRCA1 and 2 mutations. And there are some reports that say that they are offered the test, or their physicians discuss testing at the lowest level from some survey data that is out there. So that is also some of the work that we want to do is talk about it, so informed patients can discuss this with their physicians and request the test.
Yeah, it sounds like there's a couple take-home points already across all three of these presentations. One is, if you're concerned about your health risk, talk to your healthcare providers and make sure they know about your concerns, and that you can work together on making a plan for that, again, with your healthcare provider. And a second point, that the All of Us resource is really helpful for expanding beyond some older research that focused on maybe small subgroups of populations. And this giant resource that All of Us provides is allowing folks to build bridges across different groups and different populations, to take what we learn from one group and see when and how it applies to other groups, really, again, making care better for all of us, which is great. I wanted to ask another question that came in. Alex, there was a question about if you looked at genes other than APOL1 and other nearby genes. It sounds like this question is coming from the observation that you mentioned that the APOL1 allele was found initially in African ancestry and associated with kidney disease and not in other ancestries. So they're curious if you looked at nearby genes.
Yeah, thank you so much for that question. That is something that we have not done yet, but that's something that we're really interested in future studies, is looking at genetic epistasis. So I would, if an audience member has an idea of a gene to look at for that method, I would be very interested in hearing about that. So feel free to connect with me about that.
Great. And is APOL1 protective against other kinds of viruses other than HIV-1?
Yeah, it's something that we're looking into. From the statistical standpoint, what PheWAS does is essentially try to answer the question of, in people who have these variants, are there phenotypes that are more or less common? And from a Bonferroni significant level, HIV was the only virus that we found had a significant protective effect.
Great. And then Adriana, there's what I think is a little bit more of a technical question around, did you check the ancestral origin of the haplotypes of the variants you found that are specific of the Latinos and AMR groups? And I think you're muted, Adriana.
That's a fantastic question. I see that from Angelica. Thank you for that. That is actually our next.
Now it looks like.
Can you hear me? Hello? Okay, perfect. Yes, so that's a fantastic question. I'm definitely going to be doing that next. That's actually 2 undergrad students are going to be working on that project this summer. So very excited to start digging into that this summer, and hope to have an answer very soon.
Excellent. Thank you. And I'm going to ask one more question, and that's for you, Abi. Can you comment on what are some of the next steps for your research moving forward?
So currently, we have been studying on the genomics part. So we want to expand that to the other multiomics part as well, to identify the multimorbidity. So it's kind of a new field that, maybe only a couple of studies are out there regarding this multimorbidity. So we want to elaborate in our study in such a way that, so we already did in other 3 ancestries. So we want to expand that to the other subgroups. So that is one thing. And also, we want to study that in the proteomics, so that we will be able to identify some further findings there. And also the main thing, the clinical validation, that has to happen. Maybe in the future study, we will do that as well. So that's a goal.
Excellent. Thank you. Well, with that, I think we are coming up against the end of our session. I wanted to thank, first of all 3 of our presenters who did an amazing science and did an amazing job of helping us all learn about it. So kudos to you, and kudos to all of those who you worked with, because we know that this is science is a team sport. And so thank you to all of your teams, some of whom might be listening today. Thank you to all of our audience members for joining us. Up next is a 12:30 to 1 o'clock Eastern time break. And during the break, we'd love for you, in addition to refreshing yourself, to explore the scientific session abstracts and posters. And don't forget to cast your vote for the People's Choice Award. We'll be gathering right back here at 1 o'clock for session 3, which is our poster lightning talks.